hopefully.
Ok, buenos días. Muchas gracias por estar aquí. Vamos a empezar nuestro seminario de hoy. Hoy tenemos el privilegio de tener a la doctora Joana Pereira. Joana Pereira estudió su licenciatura y su maestría en bioquímica en la Universidad de Porto, en Portugal. Después hizo un doctorado en el Laboratorio Europeo de Biología Molecular, EMBL, asociado a la Universidad de Hamburgo. Posteriormente hizo un postdoc en el Instituto Max Planck de Investigación en Biología del Desarrollo en Tübingen y actualmente está haciendo un segundo postdoc en el Biozentrum de la Universidad de Basel. La doctora Joana es de Portugal y entiende perfectamente el español, por eso la introduje en español, pero ahora vamos a pasar al inglés y nos va a dar su presentación en inglés. So, Joana, thank you very much again for being here with us, and you can start whenever you want.
Thank you, Edgardo. It is amazing. It's like I understand 100%. It's like Portuguese to me. Yes. I just unfortunately cannot speak. I would love to.
I cannot understand Portuguese, so don't worry.
Yeah, we could. Yeah. Okay. I just want to. This has to disappear. Oh, okay. Yeah. Thank you very much for the introduction, and more especially for the invitation. It's a really great pleasure to tell you a little bit more about the research that I have been doing. When choosing what to speak about, I was a bit unsure what to talk, because I moved to the Biozentrum in January, and I joined a project that was already going on. I'm actually developing my own thing independently, which means that there's still a lot of preliminary data. Things are not super solid right now, and there's not a specific story that I could tell. And so I looked back at the research that I was doing on the previous life, so where I was before, that could be interesting to tell. And I really like this story, and that's why I chose it, because it was done during the COVID lockdown last year as a hobby project after work. So this was work I did with Andre Lupas, which was my postdoc mentor in Tübingen, and through whom I got to know Edgardo. And this started as an observation that Andre did, and then it contacted me about it, and I was super curious about it, and so I started playing with sequences after work, after dinner, in the couch, and it actually took me longer to write the paper than to do the analysis. And this is a story on beta propeller domains and a big superfamily called the VCBS superfamily that we looked a little bit closer at. So what are beta propellers? Beta propeller domains are protein domains that adopt a beta propeller fold. And this is a fold that has a very fascinating architecture, that it's simple and beautiful at the same time. This the structure, or this fold, is highly repetitive and is made of the repetition of a variable number of four-stranded anti-parallel beta sheets, which we call the blades, because, well, the fold looks like a propeller, you know, from helicopters or airplanes, and the units of a propeller are the blades, and so this is the same parallel. And there are many different structures of beta propellers known, which represent also a huge variety of families that I will introduce soon. And as you can, and here I have 8 examples of 8 beta propeller domains and go from 4 blades up to 12 blades. And within the different number of topologies, you actually find different families of beta propellers, and also one given family can have members with different numbers of blades, so they are super variable. What makes different families are actually the different sequence motifs that these blades carry. I have here 2 examples. One is an example of a WD-40 beta propeller, and these are characterized by a tryptophan aspartate sequence motif in position 40 of their blades. So that's why a WD 40. And this motif is a structural motif. And WD40 beta propellers, for example, one example is protein G. Another motif is this FG gap. This is example. There are many different ones. These are just two examples I'm showing. And this FG gap is characteristic of the beta propeller at the head of alpha integrins. These integrin beta propellers have this FG gap and another calcium binding motif, and this is relatively important for later in the talk. And this FG gap motif is involved in macromolecular binding. So when we look at all the beta propellers that are known so far for which proteins, for its structures exists, there are really a variety of families, and this is a list of some of them. And in this table, I also list the molecular function that these members of these families are usually involved in, as well as biological processes and the variety of topologies or number of blades that the propellers within this family can carry. And as you can see, the most common molecular function is related to binding, and this can be protein and DNA binding, small molecule binding, sugar binding, or something else. And there are also a few other families that have enzymatic activity, and these are mostly involved in virulence or pathogenic processes, but some are also involved in metabolism. So what you can see from here, these are really, really a wide variety of protein domains. They can do many different things, and what defines what they do is the sequence motifs that they carry in the blades, in the unique blades that they have. So this means that when you look at all the propellers with known structure that we know, they span a wide diversity of sequence similarities, not only within different beta propeller families, but also within the individual blades. This is what these two histograms show. So in this histogram, I compared beta propellers of known structure to each other, aligned their sequences, and calculated the sequence identity between them. And what you can see is that there are propellers that have up to 100%, I know it's this one, 100% sequence identity between them, but also as low as almost 10%. This is almost, you would not be able to say just by looking at these two sequences that they are actually homologs. And the same kind of behavior we see for individual blades in the same propeller. So this means that as soon as propellers repeat and form, then their blades can accumulate a variety of mutations, diverge a lot, as long as they keep the motifs that makes them function as that propeller is expected to. So one would expect that maybe some of these cases, especially for this, in the case of similarity between propeller families, that some of them may be occurring due to convergent evolution, because how would you be able to say that they are actually homologs? The way that we do to, or what we usually do to evaluate whether different protein family or different protein sequences may be homologs or not, is to not use phylogenetic trees, but use cluster maps, or you can also see them as a sequence similarity network. And this is basically, this is a simple concept. If you would take a bunch of sequences and compare them all to each other by using different alignment methods, the most common is BLAST. So if you would just BLAST or align them all to each other and get a statistical measure of how similar these proteins are and whether they are homologs to each other or not, you can build a matrix that carries the probability of these two sequences to be homologs based on the full set of input sequences that you have. And then from this matrix, you can see that as a distance matrix, which just tells you that, okay, this protein is this distant from that protein, and from that protein, and from that protein, and so on. And by having all of this information, you can reconstruct the position in space, of these sequences relative to each other. And then you have a map like that, where lines that connecting two dots, so dots are sequences, and the lines define whether these sequences would be connected based on the values that you have on this matrix. So the darker this line, the more probable of the sequences to be homologs to each other. And also the closest they are, the more similar they are, and they probably form a family, or depending on the thresholds, you use a subfamily or a superfamily. And so a few years ago, in Andre's group, they carried out this study, like such a study for all of these beta propeller families, and came up with this cluster map, which you can also see as a classification landscape of beta propellers. And what this showed is that actually, although they are very distinct at sequence level, these sequences are actually homologs to each other, and they're all related to each other, with some cases not being connected because they just fall below the significance values that one would expect. And so it's not really sure at this time whether these sequences are homologs or just emerged by convergent evolution. But most of the families are actually related to each other. But since then, so this work was done in 2013 and using 3,000 sequences. Since then, there were other 3 beta propellers that were deposited into the PDB, which were not included on this classification of beta propellers. And these were seen as, these seemed to be related to each other because they shared some motifs, but they didn't fall into any of the families that were described before. And as they were found in Vibrio, Colvellia, Bradyrhizobium, and Chevanella species, they were called, this group was called the Vibrio Colvellia family, or for short, the VCBS family. And so we wondered how would they affect this landscape? Where would they be positioned? To what are they similar to? And so that's what I did. I took the sequences of 13 beta propeller families, and including also these 3 VCBS beta propellers, and collected and searched for sequences in the NR database that would be homologous to that. And I collected 6,000 sequences from bacteria, archaea, and eukaryotes, and then did the same kind of analysis with these sequences. So I got the double of the sequences that they got in 2013. And when building such a classification landscape with 6,000 propeller sequences, we observed that there were 3 major groups or hubs. There's a WD40 group here on the side. There's an ASPOX group here, which is the catalytic ones. And then there's the group which includes the VCBS beta propellers. And while the WD40 and ASPOX groups were expected, this was not expected, especially because of the inclusion of, the presence of integrins and tachylectins on this group. And why? Because when we looked at the classification landscape from 2013, done with 3,000 sequences, integrins were actually connecting to the ASPOX group, which is not the case anymore. And tachylectins, there was only one tachylectin sequence back then, and it was within this big WD40 hub. Not anymore. We collected many more tachylectin sequences, and it seems that all of the intermediates that we collected had enough sequence information that actually placed them in the right, well, we think, the right position within the VCBS group. So as conclusion from this analysis, we observed that the VCBS-like propellers form a third hub, which is clearly delimited from the other two major groups that were already known. And the second is that integrins, so the beta propellers in integrins are members of these VCBS superfamily. So they are distant relatives of these three other members that were solved or seen after the study from 2013. And funny enough, this relationship was previously conjectured when the different structures of VCBS propellers were solved, but they were regarded as convergent evolution because there was not enough sequence similarity between them. And the third observation is that also tachylectins are not members of the WD40 family, and they are not singletons. It was believed that they were singletons because there was only one sequence known, but actually there are many homologs in other different species. And also, as with the integrin-like propellers, this relationship between tachylectins and VCBS beta propellers was previously conjectured, but also discarded due to lack of significant sequence similarity. And why were we now able to see these relationships that were not seen before? This is because we collected, by doing blast or PSI-BLAST over a huge database of sequences like NR, looking for homologs of our starting sequences, we collected from a variety of species, especially metagenomic species, a variety of intermediate sequences that formed the bridge that connected the different families that we know. So we found all of the intermediates at sequence level or at the sequence space that started to make little bridges between each other to form a huge bridge that then bring things together. So this was quite of a really nice message, and to me is also one of the major messages of this work is that by having a much larger set of sequences and the exponentially growing database of sequences coming from all of these metagenomic studies, you start having all of the intermediate sequences that are allowed for a given protein family that bridge families between themselves. And you can see more clearly evolutionary relationships between different protein families. And what brought these families together is the presence of these motifs that are specific to these kinds of sequences. And there are two motifs. One of them is this cation binding motif that is also conserved in alpha integrin beta propellers, as I introduced a few slides back. This motif is conserved within the entire VCBS hub, except for the tachylectins and the PQQ, these are the sequences that bridge VCBSs to WD40 beta propellers. And this is a conserved motif that has a D, then a G, then a D, then a G, then a D, then a G. So it's a DG, DG, DG motif that from structures of propellers from these, like this PVL and the AU, DH and the DCB, we know it's involved in cation binding. So it's expected that in the other families that we found, it's also involved in cation binding. Which one, not sure. In some cases is calcium, in others is zinc, but so it's not easy to say which metal would be, but it's at least a cation. And the other motif is this GW at the end of the blade, and this GW is present in VCBS propellers that are involved in binding binding sugars, so in lectins, like this PVL, and we also see it in tachylectins and some intermediate cases. So these are the two concept motifs that bring all of these PVL integrin. So you can see in integrin you have the cation binding, and in PVLs you also have a cation binding. And in tachylectins you have the GW, which is also present in PVLs, which is one of the core VCBS's families. And so when we map the presence of these motifs into the landscape, it's kind of like you can see motifs being gained and lost. So this is this is hand waving, but I like to just think that we can see this. It's not proven that there's actually these processes of losing and gaining, but it kind of is like that. So if you start at the WD40, which is in that direction, and you come to this PQQ, which is the place where you start the connection between all of the other. So this is kind of at the center between all of the hubs. You have these PQQ, which are here and have none of the motifs, and then going in this direction. So the bridge, these RGL11 PQQ bridge, is these sequences here. You start seeing that the motif for cation binding starts appearing in the sequences. And so it is kind of like exiting this family and coming in the direction of this bridge that connects here. You have the gain of the cation binding motif, and then arrive here, which is called the VCBS core, or I call the VCBS core. Then here, many things happened and take these superfamily into different directions. In this direction, you have the divergence into integrins with the gain of the FG gap motif. And so it comes here, the integrins from prokaryotes that then duplicated to, or diverged to give origin to the eukaryotic integrin beta propeller. And here you have the loss of the cation binding motif, giving origin to these AUDH. And these, the cation binding motif, it's kept, but there's other gain of other motifs, and also some divergence of the blades that give origin to these protein binding beta propeller family, which is found in some toxins. And then here you have the side where this GW motif that is involved in sugar binding is gained. And so here is the lectin side of the VCBS beta propeller VCBS cluster. And then going in this direction, there's the loss of the metal binding motif, and the kept and the sugar binding motif is kept, giving origin to the lineage of tachylectins. What is also interesting when I look at these set of sequences is that the ones from the so the original VCBS families are actually at the periphery of this big blue blob, which is this AUDH, this TCB, and this PVL. Here in the middle, there's a bunch of sequences that are called hypothetical proteins. This is the core VCBS sequences, and these are the ones that are responsible for making most of the bridges between PVLs, integrins, and tachylectins. These were never mapped to VCBS, and when I look at the domain environment where these beta propellers are found, they are found in a variety of domain environments representing a variety of novel protein families that are just called hypothetical proteins. So there's a huge potential here of proteins that we know anything about, that have these likely lectin or lectin-like beta propellers and domains that are involved in binding, like the immunoglobulin domains and the armadillo repeat domains for the jelly rolls, but also some catalytic ones like these flavodoxins here, and other things here like this. These are peptidases also. And some of them have a transmembrane region or a signal peptide. Actually, is a signal peptide. So it's probably they are secreted. So these are things that these species may be secreting, but we have no idea what they do. They are completely novel protein families, things that were in the dark that was just highlighted like that. So I found this really, really fascinating. And these stars mark which families are new. And so you have stars everywhere. Everything's called hypothetical, everything would be new. And they are not specific to a given taxonomic group. They are actually widespread, mostly in bacteria, but you can find them in all different families. So these numbers define the different clusters, the different families, and then they have them here. And this is a taxonomy which phylum I find within the sequences, and or where in which phylum I found the sequences, let's say. And you can see that I have many phyla represented there, and some are more specific to some phyla than others, and others are found everywhere. So yeah, there's a huge diversity of things. And this is not only in bacteria, there's also in archaea and also some eukaryotes. So for example, this is not novel, this is integrin, but within, for example, this tachylectin-like, which, remember I said they were thought to be orphan genes, just be one. Actually, they are found in bacteria and in eukaryotes. And I'm especially interested in these because there are some very interesting classes here. So the original tachylectin, it's this cluster here, and which the one that was thought to be originally an orphan. It's this 9, and you can see it connects to this, which is in bacteria. So this is the beta propeller, connects to this, which is in bacteria. This one, which is also in bacteria, especially this is specific to actinobacteria, as well as this one, while the 8 is kind of widespread. So this would be the tachylectin-like that are more similar to the lectins from the VCBS lectins, and these carry also the cation binding motif, and then it lost it and it gave origin to this bacterial one that then in actinobacteria a novel family emerged, and this also gave origin to the eukaryotic one. So this would be interesting to study further to understand actually what is the origin of the eukaryotic one. How come something that is quite widespread in bacteria now is found in specific group of crustaceans? And so as just a little bit background into tachylectin. So tachylectin 2, which is the tachylectin that adopts beta propeller fold, it's a lectin involved in the innate immunity of the Japanese horseshoe crab, and is found only in some crustacea and cnidaria. This is what was known before we started this work. And so it was believed to be a singleton because it also is also highly repetitive, which means that this suggests that it has a very recent origin. The blades still didn't have time to accumulate mutations and start diverging. So it was just thought that emerged in these lineages, and to be a specific protein that is specific of the innate immunity of these crabs. But actually, our results suggest that actually tachylectin may have a bacterial origin. Which is this origin? I don't know. That would be something really interesting to study. But at least there's homologs, very close homologs in bacteria. And in actinobacteria, there are at least 2 different families of tachylectin-like beta propellers. And so then I looked at these a little bit closer into this cluster, this specific one from actinobacteria. I was a bit curious to see what it could be doing. And for this, I used a tool that I'm working on, which is called GCSNAP. I started also developing it during the COVID lockdown. The goal of this tool is you would give a bunch of target sequences that you're interested in. Could just be UniProt IDs or NCBI IDs. And what it's going to do is to compare the genomic context of the genes that encode for those proteins. And so with this, you can try to find probable operon-like structures and get some insights about the genes that are around the genes encoding for your proteins of interest. And what it has different from other tools of the gender is that it's very interactive, and you can click on the results and be directed into either structural databases like the Swiss Model Repository, or to sequence databases like NCBI. And so this interactivity makes it much easier to interpret that. And so I ran this tool for this cluster 10, and I got that most of the genes encoding for the proteins in this cluster are very close to this very long gene, which protein product is predicted to have a signal peptide 2 as the propeller itself. And this seems to be a conserved gene. You can find it in many different species. So I was wondering, well, what is within these? Because it seems the protein is going to be super huge. And this was before AlphaFold. And so I was trying to use the state-of-the-art methods at that time, which was homology modeling and Rosetta, to try to model with some grasping by myself, like for this case, and got a really interesting protein that has an N-terminal domain that doesn't fall like anything I know, 2 immunoglobulin domains, then this very huge cocoon, which has a beta sandwich insert domain somewhere, which I don't know where it goes. Then there's a super helical domain here, then an intane domain, and then a sulfy redoxin domain at the C-terminal. And then there would be the tachylectin. Now, interesting enough, such cocoons are usually found in secretion systems and toxic secretion systems, and I wonder what these big protein may do, especially when it has an intane domain, which a hint domain, intane, which suggests that there's some kind of post-processing of it. It probably clears itself to release something. So we may have uncovered here a very interesting system, so I wonder what this thing would do. And so from this work, we uncovered a lot of different things, and it was just a small bioinformatics analysis that highlighted so much gold and so many interesting things. So the first is that, well, the VCBS beta propellers are actually a very large superfamily, likely binders, that are found in most kingdoms of life. There are many VCBS families that are in hypothetical proteins that were never described before. That the beta propellers in alpha-integrins and tachylectins are members of this superfamily. The tachylectin 2 may have a bacterial origin, and its ancestor lost the cation binding motif. And the actinobacteria carry at least 2 unique tachylectin-like beta propeller families. And for me, the most important take-home message of this study, something that I already said in the beginning or in the middle of this talk, is that this was only possible due to the large amount of data that results from large metagenomic studies. And this just highlights that the higher the amount of sequence and structural data that we have, the better we can predict and understand protein evolution and function. And so with this, I would like to thank, of course, Andre for his immense support and really a nice mentorship. I really like to work with him, and we always did very, very interesting projects together. And of course, the department in Tübingen and also to now my current hosts, the Professors Veda group, which is the computational structural biology group here at the Biozentrum, which gave me complete freedom to explore more what is hidden in all of these hypothetical proteins in a very large scale, and continue developing GC-SNAP and the other tools that I started putting together in the past years. So yeah, with that, I am finished, and thank you very much.
Thank you very much, Joanna. We will have now time for some questions. If somebody wants to make a question, you can either write it in the chat section so I can read it to Joanna, or just raise your hand, open your microphone, and ask it. In the meantime, while people get bravery to ask, I want to please ask you very strongly to please develop that GC context tool. Right now I'm suffering because the normal tools that we used to have to study gene context in at least in bacteria, the one from Mexico is broken, it's not working anymore. You can only get now what they have in their database that is very limited. And the one from the Netherlands just died. It just disappeared. I don't know where it went. It's gone. So yeah, please do it.
Yeah.
And when you have it, send me the link.
Well, so GC, so the tool GC-SNAP is available to download on GitHub, on my GitHub page. So this is a terminal run tool, but it's very simple to use. So you just need to set it up. It's Python based. It's very easy to set up, and then you can run it in your computer. And it's another thing that it's also really nice about it is that it is super flexible. You can have any, as long as this genome of the species is on GeneBank, it will do the analysis for you, because it doesn't rely on pre-computed data. It does all the searches on the go. It does collects the data for you, and then gives you the visual output that then you can explore and play with it. Yes. And this is something this is a project that I took with me, so I'm continuing developing here, and so just stay tuned, but you can already play with it. It's on
cool. Thank you very much.
And there's a paper. So if you search GC-SNAP and my name, you get a paper, and from there's the link for the GitHub.
Okay. Thank you. We have a question here from Rommel. How, Dr. Joanna, very interesting presentation. Related to the last comment, what is your opinion about the use of AlphaFold 2 and its role in amplifying protein structure databases?
AlphaFold is really good. It's very accurate. Our experience with it is really accurate, and the models that come with it are really good. They have now also ways of modeling complexes. I don't know how you if you have been keeping updated about what has been happening, but the field has moving so fast since they released their code, and in a few months, like in 2 months, they improved their method to also do prediction of complexes, and it's also really accurate. The only thing that they are not able to predict so well is actually interactions with antibodies, but the other things, they are super accurate. And so I'm really excited of how the field is changing, and they also say, so they released structures for all of these 21 proteomes, but they promised that by the end of the year, or in the next few months, they are going to model structures for UniRef 90, which means that this is basically all proteins that we know now. So we're going to have models for all proteins we know now. And so it's not like the field of structural biology is going to end. It's just a complete novel start of the field. And I am very excited about it. And just stay tuned with it. And yeah, yeah, my opinion about the use of AlphaFold is if you have the chance of using it, use it. I think it's going to change the way that we do research. Before, we would start hypothesis and then solve a structure, and then from the structure do something else. And now you're going to start always from a structure that will help you make even more informed predictions. And you can still go to the lab and do structural biology, but you will always have a much more robust starting point, let's say. And so, yeah, that's my opinion. And the role of amplifying protein structure databases, well, they will, because they will solve, or they will predict structures for UNIREF 90, and they will make this publicly available to the community, to everybody. This is not going to end up in the PDB. This is going to end up in a dedicated database. And so, yeah, but you can still see the structures in UniProt, or you usually are linking it. So yeah, I hope I answered your question and just didn't babble around.
And he made a follow-up question. Also, if your work pipeline can be applied to any other protein families.
You mean like this building these cluster maps and the idea of the follow-up? Yes, definitely. This is an example of such a work that we did, but we also did these things for other families. There's the example of we studied an RNA binding domain where we found a third family. We also did this for ultramembrane beta barrels, and these are just two examples. Actually, if you search Andre's papers, they always start with such a cluster map.
Maybe we need to publicize right now the bioinformatics toolkit from the Max Planck Institute from Developmental Microbiology. Just biology, just search for it. Just search for Max Planck bioinformatics toolkit, and maybe Andre Lupas. And with that, you will find a platform, a web-based platform that is very useful for non-bioinformaticians like me. And it's like the front end of everything that Joanna can do with her hands. For us that we cannot do it with our hands, we can do it with our web browsers using this site. And the CLANS program is there.
Yes.
It's very easy. Someday I think we were trying to bring Vikram to teach a small course on this, but the pandemic canceled it. But we will try to do this in the future.
Yeah.
Okay.
I think CLANS is a really nice tool. There's an updated version of it now, which works a little bit better. But yeah, as you said, the toolkit is the front for all of these tools.
Yes. And finally, Rommel said, "Excellent, thanks, and I wish you success in all your projects."
Thank
Now,
you.
Ramon is asking, your pipeline is only taking in account classical gene model for finding genome context?
It doesn't use any model. Basically, it just given your input targets, it goes into GeneBank and looks in the genome where that sequence is annotated to, what are the genes that are flanking it, and takes them out. And then runs such a similar sequence similarity searches to find groups of genes that codify for protein sequences that may be homologous to each other. And then colors based on that. And then it's up to you to do interpretation. It doesn't tell you, oh, this is what I think is going to be an operon. It just shows you what are the genes that are there and how similar they may be, and then gives you some not statistics, but basically, for example, one of the things that it shows is at each position, what is the most conserved gene, or what is the mostly found gene. So in the middle, you always have your target, and then in the 3', the most found gene is one that codifies for proteins that seem to be from this family. But this is just what he sees. then it's up to you to make interpretation. So there's no models of anything in there.
Thank you. Jordan asks, hi, Dr. Joanna. Thank you so much for your presentation. I found really interesting how you did the wall analysis from available data just from database. I did not fully understand why you choose the VCBS family. Was just because it was not studied before, or there was another reason?
It was just because it was not studied before. This came as yeah, a hobby project. We realized, oh, there's these structures. They are not in this cluster map. What happens if we put them there? Voila.
Yes. And something they may not know is that Andre has this special power to see patterns, no? And suddenly he notices a pattern, and then he shares with his people, and then we can build something from these patterns he notices, no? This always amazes me from him. We have another question, a follow-up question from Ramon. I think he deleted it.
There's one.
Ramon,
What
about the codon usage? Is it taken in account?
No, it's not based on genomic sequences. It's really just based on protein sequences. Yeah.
Cool. Are you still there? Because you froze.
I froze. I'm here.
No, I froze. It was my problem. Sorry.
Yeah.
Okay. We have another question from Jordan. Also, besides the VCBS family, is there any other family that has not been analyzed as you did with VCBS? And if so, could this clustering change?
Well, I don't know if there's any other family. Maybe I didn't keep up to date to the new families that are described. But there's a chance that yes, and if included in the clustering, yeah, there's a chance that things could change. These cluster maps are very sensitive to what is in there.
Yeah.
And so if you put something that now it's a bridge between families that were not connected before, yeah, it's going to change the landscape. But this is what makes it really interesting to do this, to update these cluster maps constantly when new data comes in. Yeah.
Ramon is also very excited about your tool, I think. He's asking now, really, there is a lack of tools like this right now in the market, as to say. He asks, this pipeline could help to find genes related in different positions in the genome, or just the closest genome context?
So, yes and no. Yeah, it's basically a yes to both things. So this is just, it shows you what is conserved within that given window. So imagine that, yeah, you have your targets, right? And you want to look at what is in the neighborhood of each of them. That's the first thing. And so for each of them, you look within a window, and then just take all those windows and align them based on your targets. And so if something is conserved within this window, they will be highlighted, even if in one case it's here and in the other case it's on the other end of the window. So it's always what is conserved within that window. If you have one window that has a gene, and then that gene is conserved in another genome that you have represented in the data set, but it's not within that window, it will be somewhere else, that's not going to be shown. So I hope was it clear?
Yes, I think
okay,
so.
so he's next to you.
No, somebody called me. No. And I also have a question regarding this. For example, if you remember, I work with very big proteins, and these proteins have multiple domains that are related to some other proteins. So, for example, to importer proteins. So, if we run the search, will we found the importers alone and my big proteins with the importer domain, or will it only found the importers?
You mean the GC snap?
Yes.
So the way that it's done now, if you have, no, it will so if you imagine that you have a fusion protein, right? And then, and in other species, you still have the fusion, and then in other species, you have the two separated. These three, so the fusion and then the A and the B, they will be seen as three different things.
Cool. Good. Super. Thanks. I'm sorry for asking about other stuff. Oh, we have another question.
Sure. No, go ahead.
Well, it's a very long question from Giovanni Ruiz Romero. Thank you for this very interesting study. My question is, what is the molecular or structural explanation of the specificity of beta propeller domains to bind of a wide different structures like proteins or other metabolites such as sugars? I ask because of the wide variety of functions related to this domain, such as ATP production or cell signaling.
Short question is, I don't know. But I think that this is such a special scaffold. So if you look at beta propellers and you make like a surface representation, they look like donuts, which means that they are flat on both sides. They have a hole in the middle, and then they have a huge surface like that. And so this has a very large surface accessible to different things. And so this is basically a really nice scaffold to start putting small structural motifs that would give you specificity. Some propellers, they bind proteins by the top of the donut. They just glue it to it. Others have catalytic sites in the donut, in the hole of the donut. The ones that bind sugars, they bind sugars around the donut. So I think it's just that they are a really nice structural scaffold to accumulate small structural motifs that allow for these different functions. But this is me hand waving.
I remember when we talked about this with Andrea and Amit, you remember he was trying to purify some of these proteins. The explanation Andrea always just was surface. You have a lot of surface, and then you have a lot of space to set interactions. No, maybe it's the same you said. And finally, I think we will stop now. One more question about your pipeline. Can I run? It's not mine. Can I run the script in a local computer?
Yes. So the requirements are explained in the GitHub page. I run it in my small MacBook. It runs. You can define how many CPUs you want to use. There are some specific things that you have to have installed in your computer, like Blast should be installed on your computer. And I think it's mostly Blast and Python. You need Python. And then when you install GCSnap, it will install all of the Python models that are necessary. But if you run into any problems installing it, just drop me a line and I'm happy to help. Especially if you have a Mac, it's really easy for me to solve. If you have a Windows computer, it's a bit more difficult, but I make an effort.
Ramon says, sounds really nice too. Thanks. Yeah, we're really excited. Just think about all the citations you will get, Joanna. keep working on it.
I will. Thank you.
Okay, so if there's no more questions, I think we're going to end it here. Gracias, Joanna, once again for being here with us for a very nice talk. I will email your diploma or your yeah.
I have a certificate.
So you can put it on the wall in your office. And yes, see you next soon, hopefully, when things get better. Yeah. Adiós. Gracias.
Bye.
Bye. Oh, they are giving you applause.
Gracias. Buen día. Muchas gracias.
Gracias.
Thanks a lot.